Thanks a lot, David and Carl, for inviting me. I'm really excited to be here, part of this series. And I'll tell you a little bit about the work that we do in my lab, where we are interested in understanding visual processing, in particular, visual processing of natural visual inputs, like this kind of image that I put on the screen. And our research in the lab focuses on three interrelated areas and sets of questions. One is understanding the activity of neurons in the visual cortex of the mammalian brain. So how neurons respond to visual inputs, how they interact with each other, and how they transmit information. And we do this work in collaboration with experimental labs that can record and manipulate, perturb the activity of neurons in the visual cortex of monkeys and mice. We are also very interested in visual perception, understanding the strategies of human visual perception and the algorithms that we use to sort of solve the complex real-life tasks that we continuously solve when we are in the process of so-called natural vision. That work also involves collaboration with experimental labs to run psychophysics experiments. We also run some of those in my own lab. But the core of the work that we do in the lab really is to develop theories that can help us understand this rich phenomenology, both at the neural level and the perceptual level, and hopefully also understanding the link between the two. And the framework that we've been using, which I will talk about a lot today, is the framework of probabilistic inference. So most of you will recognize this equation here. And the reason that we focus on that framework is that it really provides us a perspective and a conceptual and mathematical way of understanding how to deal optimally with the main challenge that the visual system and sensory systems in general face. That is the fact that the inputs are noisy, just because of the biology of the system, and they're ambiguous, meaning that there is not a simple one-to-one map between objects in the world, physical objects in the world, and the sensory signals that we receive. And so to deal with that noise and ambiguity, probabilistic inference provides a framework to understand how to do that. And so that's the framework that we will use. So before I start and go into the details of our work, and I should mention that our focus has been on neural variability and perceptual uncertainty, because those are two sets of phenomena that really tie into this idea of probabilistic inference. And I will discuss that later on. But before I talk about our work, I just want to spend a couple of minutes to contrast that with more traditional, long-standing approaches to describing the phenomenology of visual cortical activity and perception. And so I'm sure you're all familiar with this kind of parametric statistical models, linear, nonlinear, Poisson, and similar models, which have been extremely successful in quantitatively describing the responses of visual neurons to simple stimuli like gratings or moving dots, with this conceptual separation of a simple linear filter, maybe a nonlinearity to account for the transformation from membrane potential to firing rate, and some sort of noise or stochasticity to account for observed variability in spike counts. So these kind of models are very successful, although it's been recognized that they kind of hit a wall when you try to use them to predict the response of neurons to natural visual inputs. And then in more, in the last five to 10 years, there's been an explosion of methods that kind of go beyond this simple single neuron LNP, leveraging these advances in machine learning, in particular deep convolutional neural networks, which now have lots more free parameters, but there are also very smart engineering techniques to fit these parameters, not directly to neural data, because there's not enough of them, but to image data to solve tasks like object recognition. And this class of models now provides us with a quantitative ability to quantitatively predict the responses of neurons throughout the visual cortex to natural visual inputs to some extent. So that's a success story, and it sort of provides us with foundational knowledge and understanding of the selectivity of neurons throughout visual cortex. It also has some limitations. This kind of encompasses the traditional view of visual processing as a feed-forward cascade. And so what we like instead to take a complementary approach that builds on what we call the normative approach that uses normative models. By the way, do you see the title of my slides, or do you see the control panel up here?
We see the title of your slide.
Oh, okay, because I have a control panel here, so I wasn't sure what you're seeing. All right, so so we want to take this complementary approach that uses normative models. In particular, we want to start from the idea that the computational goal of neurons or neural circuits in visual cortex is to build optimal, in some sense, optimal representations. And we'll focus on probabilistic inference, as I mentioned earlier. And the idea is that this framework of hierarchical probabilistic inference can give us traction to start understanding how neural activity varies over time and how that variation is coordinated between neurons. And more generally, that this framework of probabilistic inference can help us understand the role of recurrence and feedback, which are heavily conserved anatomical features of cortical organization, the role that they play in solving complex computational tasks that we face all the time. So this is the general framework that we take. So I'm going to tell you briefly about some of the work that I did as a postdoc with Adelia Schwartz to kind of set the stage and introduce a couple of key concepts. I will then spend most of the time of the talk on this second point here, where I'll tell you how we're trying to develop a unified theory of contextual effects on cortical variability and response statistics. And then if there is time, I might tell you a little bit about the work we're doing on perceptual grouping and segmentation. So as I mentioned, this first part of the talk is going to be a very, very brief recap of the work that I did as a postdoc with Adelia in collaboration with Peter, Diane, and Adam Cohn. I'm listing here a few publications for those of you who are interested to go and look at the details. I will not give you all the details now, but just again, the key ideas. First, before I go into our own work, I just wanted to provide an example that I'm sure you're all familiar with of a successful normative approach. So just to repeat again the distinction between the descriptive parametric model fitting approach and the normative approach. So I'm sure many of you are familiar with this work on sparse coding by Bruno Sausen and Belensinowski and others, who showed that if you assume that the goal of neurons in V1 is to build a sparse representation, an efficient representation, sort of saving on metabolic expenditure to represent initial inputs, then what you get is this kind of receptive fields that are filters that are selective for orientation and spatial frequency and are localized in space, and that qualitatively capture the well-known tuning for orientation of visual neurons in primary visual cortex. And the interesting thing is that this is a parameter-free prediction in a way, right? This principle of sparse coding applied to the statistics of natural images generates some predictions about normative predictions about how neurons should respond to inputs without ever using any specific neural data. And so this is the kind of normative approach that we want to take. And our goal was really to go beyond sparse coding in one way and to go beyond orientation selectivity, to look at this sort of a more nonlinear set of phenomena in primary visual cortex that is termed contextual modulation, of which one specific example is here. This is an example of surround suppression. So this is a phenomenon where if you present a stimulus inside the receptive field, here the blue dashed circle, and you increase the size of the stimulus, what you observe is initially an increase in the response of the neuron in firing rate, And then once the stimulus goes beyond the receptive field, you have a reduction of responsivity. That is called surround suppression. And it's a nonlinear phenomenon because the contextual stimulus by itself does not drive the neuron, but when it's combined with the stimulus inside the receptive field, it produces this sometimes strong suppression of responsivity. And so our goal here was to develop a set of models that would start from this computational principle, that is assuming that the goal of the neurons is to perform probabilistic inference on the visual inputs, and to see to what extent that could capture contextual modulation, both generally and specifically with a set of specific predictions. So to do that, we needed, first of all, an understanding of the statistics of images, so that we could translate that into a probabilistic generative model, which is what you need in order to perform probabilistic inference. And so these are the three elements that I'm going to describe very briefly in the next couple of slides. So first of all, the statistics of natural images. If you take filters like the ones that you obtain from sparse coding, these kind of oriented localized filters, and you apply them to natural images, what you get, of course, is a sparse distribution. But it's known that the level of sparseness, it can vary from region to region of natural images. For example, in high contrast areas, you'll have more of a range of activation of this filter as opposed to a low contrast area. And one very fundamental property, statistical property of natural images is that neighboring filters are informative about the sparseness of each other. So in other words, if you look at the activity of these two filters across natural images, you'll find this statistical dependency that is visualized as a bow tie shaped conditional histogram, where the magnitude of activation of one filter predicts the level of sparseness or the variance of activation of the other filter. And so again, you could think of the magnitude of one filter as a rough indicator of the contrast level of the image in that region of the image, and which will then predict the range of activation of the other filter around that location. So this, the lab of Moncelli introduced this very simple statistical model, a probabilistic generative model, which starts from independent local features, GKGS here, and generates this spatial dependency that is typical of natural images simply by multiplying the independent local features by a common what we call global modulator, this V variable here, which is shared between across features. So this scaling common scaling factor gives the name of Gaussian scale mixture to this kind of models and generates this dependency. So what we proposed when we started working on this was that we could assume that the goal of neuron CB1 is to, given the visual inputs, you could think of these as the feed-forward synaptic inputs, perform probabilistic inference and estimate something about these latent features. That would be the goal of a neuron in B1. And so in this early work, what we did was to focus on the expected value of this local feature in the posterior distribution. And that requires solving some integrals and marginalizing out the global modulator. And at the end of that process, you end up with something like this expression, where the expected value is a ratio. And in the denominator, you have some sort of measure of the energy in the image in the center receptive field of the neuron and in the surround around the receptive field of the neuron. And again, this can be derived analytically, but the intuition is very simple because the generative model is multiplicative. If you observe K and you want to infer G, you want to do something like divide K by V. And because you do not observe V, but you can estimate it from some combination of K and S, from the energy in the center and surround, then you end up with this, approximately with this division. And of course, you can see easily how that expression qualitatively reproduces surround suppression. When you increase the size of the stimulus, initially you increase only K, so both numerator and denominator, the activity goes up. And once the stimulus is outside the receptive field, the only thing that changes is S, which is in the denominator. So you increase the denominator and you reduce the activity of a model neuron in this simple model. So qualitatively, inference in the GSM can reproduce surround suppression. And again, as I said earlier, we had a series of papers where we explore in great detail different properties of surround suppression, how it's tuned, how it depends on the neuron's receptive field properties, and so on. I will not go through those, but the only one additional thing that I want to mention is that of course, this GSM, Gaussian scale mixture model, is an oversimplified model of natural images. For example, one important aspect of images is that if you're looking at regions of an image that corresponds to different physical objects, you would expect that the activity of filters applied there are less statistically dependent. And in that case, a better model is one where there is not a shared modulator, but rather some sort of more local modulators. And the prediction from this, again, I'm skipping the analytics here, but the prediction intuitively was that when the inputs are what we call statistically homogeneous, then you have a prediction of surround suppression. But when the inputs are statistically heterogeneous, that is not dependent, then you would predict that there is no contextual modulation nor surround suppression. And so to, long story short, we went to the lab of Adam Cohn. We designed some experiments and collected data to test this prediction. And what I'm showing here is one example neuron where you can see that for the images that were homogeneous, there was a strong surround suppression, the ratio between the response to a large image over a small image. But for the images that were heterogeneous, there was on average no change of response for this particular neuron. And this was true across the population of neurons, with the majority of neurons showing a larger suppression from homogeneous images. So the important thing here was that this model, which we call the mixture of GSMs, was never trained on neural data. This is a parameter-free prediction of the strength of surround suppression that is only dependent on the assumption that neurons in B1 perform probabilistic inference by inverting a mixture of GSM models. Lastly, this is work that does not involve me or my lab, but I wanted to point out a paper that came out last year that kind of combined detailed models of recurrent excitation inhibition and experimental work with recordings and perturbations in the labs of Ken Miller and Massimo Scanziani, which identified a sort of a cortical microcircuit that implements this kind of flexible modulation, where specifically the excitatory neurons for which we're studying their flexible surround suppression are surround suppressed primarily through this class of inhibitory interneurons, somatostatin interneurons. But then there is another class of inhibitory interneurons, the VIP interneurons, that primarily suppress the somatostatin interneurons when the stimulus configuration suggests that there should be no surround suppression. And so I like to bring this up because I think this is a very nice example of how we can go full circle, starting from this normative principle of probabilistic inference, derive some very specific predictions that we could, for cortical activity, that we could validate with electrophysiology, and then take it one step further and really use these findings and these predictions to constrain mechanistic models of the cellular and circuit mechanisms that could implement that computation. So that's a nice story. One thing that is being left out in everything I've told you so far is variability and how that variability is shared between neurons. So this variability of neural activity, as I'm sure you know, is a widespread in cortex and is a signature really of recurrent dynamics in balanced or roughly balanced EI networks. And so really we think that in order to link mechanisms to computation, we need normative theories that can go beyond just predictions for firing rate. And so this is what I'm going to discuss in the second part of the talk. This is work that is ongoing in my lab, primarily done by a former postdoc, Dylan Festa, and my current postdoc, Amir Farzmadi, and student Oren Weiss. And again, this is work done in collaboration with Adam Cohen. So we have discussed surround suppression as an example of contextual modulation in V1. Just to, it's important to point out that that's not only a quirk of V1. Contextual modulation is widespread throughout the visual system. It's also been observed in other sensory modalities, in multisensory integration, and even beyond sensory processing in value-based decision making. We have a plethora of examples of how contextual information modulates the activity of neurons. There is another, as I was saying, widespread cortical phenomenon, which is response variability. Here illustrated with this plot of the variance of the spike count across repeated presentation of the same stimulus as a function of the mean of the spike count. So each point here is the variance and mean of the responses of a neuron to repeated presentation of the same stimulus. And this variability is not unstructured. It has structure. For example, there is a relationship between the mean and the variance. It's not exactly Poisson, which would be the red line here, but there are some deviations from Poisson. The variability is also coordinated between neurons, is correlated over time, and it has many properties that have been studied extensively for a number of reasons. And this variability, again, is not a specific quirk of V1. It's actually widespread throughout different cortical areas, in visual cortex and beyond. So our goal in this project was to come up with a normative account of contextual effects and response variability and covariability. And our hypothesis, driving hypothesis, was that those are signatures of a probabilistic code optimized for natural image statistics. So we can come back now to our simple Gaussian scale mixture model and see how we can leverage this framework to try and understand how contextual modulation and response variability are related to each other. I mentioned earlier that the hypothesized goal of neurons is to represent this posterior distribution, given the inputs, posterior distribution of the latent features. And it's important to realize at this point that really it's not enough to just compute this distribution. You also have to make some assumptions about exactly how neural activity represents that distribution. Now, there are a number of theories for how the activity of neurons might represent probability distributions. For example, combinations of neural activity could represent the parameters of a distribution. Those are sometimes called probabilistic population codes. The hypothesis that we followed instead, that's been introduced by others, is this idea of neural sampling, where you could really conceptually think of this two-step process where, given an input, you compute somehow a target, the correct posterior distribution, and then the activity of neurons represents samples from the distribution, such that the histogram of neural responses over time or across repeated presentation of the same input will be a direct approximation of this distribution. And so there is a sort of a generic prediction here, which is that the variability, the spread of this histogram, should really reflect uncertainty in the posterior distribution. So that, for example, if you have an image like this one, where there is less uncertainty about the strength of a, let's say, a vertical feature in the image, then that should correspond to less response variability. There is now experimental evidence for this sort of generic prediction. But what I want to do is to use or leverage our GSM framework and our understanding of contextual phenomena to give you much more specific predictions and therefore have more stringent tests of this theory. So we talked earlier about how inference in this model can reproduce surround suppression of firing rate. What we did here in addition was to derive an expression for the variance of the posterior distribution. And there are a few interesting features that come up. One is that the variability also has a divisive form, similar to the response strength. So you immediately see that there's going to be a relationship between the variance and the mean, qualitatively consistent with the phenomenology of response variability. In addition, the scaling of the response variability is different from that of the response mean, accounting for some of these deviations from Poisson-like or constant Fana factors. The divisive form is also important because, some of you may be familiar with the divisive normalization framework, which is a very successful descriptive model of contextual modulation. And it's a model for which also there's been a lot of work on understanding the mechanisms of divisive normalization, in particular, how that could be implemented through recurrent excitation and inhibition. And so the fact that we find that divisive normalization influences both the strength and the variability of neural responses as a consequence of implementing probabilistic inference gives us also potentially a link to mechanisms, the circuit mechanisms underlying this computation. So those are all, I think, interesting theoretical findings and quite general, quite broad, but there's in addition a very specific property of these equations here, and that is that the surround stimuli only modulate the denominator in these equations, both for the mean and the variance. And so that conceptually corresponds to the fact that if you present a surrounding stimulus, a contextual stimulus, you provide more information about the image features, and that reduces the uncertainty. And it should, as it's expected according to this theory, to reduce the variability of the responses of neurons. And so this is something that we can test directly, and it has an advantage compared to, for example, changing the contrast of the stimulus, which is another manipulation used to modulate the uncertainty, that in this case, we're not really changing the sensory drive to the neuron. We're only changing the contextual information. So we're only manipulating uncertainty. So to test this, again, we went to the lab of Adam Cohn and we designed experiments where we record with an electrode array and we present stimuli either at roughly the size of a single receptive field or extending well beyond the receptive field to engage the surround. This is an example cell where you can see that across images, on average, there is a stronger response to small images than large images. So this is surround suppression once again. But we also observe that the Fana factor, the ratio of variance to mean, is suppressed by the large images. So there is a reduction of variability by stimulating the surround, by providing contextual information, and the reduction of variability is stronger than the reduction of the mean. This was true for this particular neuron and across the entire population that we recorded from. Now, there are a number of we had to consider a number of possible confounding factors and run a number of control experiments and control analysis. I will not go through all of those. They're all in the paper if you're interested. But one that I want to bring up is this fact that because of the design of these experiments, it's possible that the stimulus, the small stimulus, was not perfectly aligned with some of the receptive fields of the neurons that were recorded from. And so if that were the case, then this reduction of variability might be simply a consequence of going from the spontaneous state, spontaneous activity of the neuron through to the evoked activity by a sensory stimulus, right? This is a well-known phenomenon, not only in visual cortex, but also in other cortical areas, that when you, that the presence of stimulation quenches neuronal variability. So to rule out that possibility, what we did was to run these control experiments where we progressively increase the size of the stimulus. So we measured these area submission curves with natural images now. And we again observe that the firing rate goes up and then it goes down after the stimulus becomes larger in the receptive field. But we also observe that the benefactor, the variability, is reduced monotonically. So the larger the stimulus, the lower the variability. And so there is a suppression of variability for small sizes, which correspond to this well-known phenomenon of, stimulus onset quenches neuronal variability, but there is an additional suppression of variability that is purely due to the contextual stimulation. And in addition, we could also compare conditions with smaller and larger stimuli than the receptive field that had matched firing rate. And again, we found that the larger stimuli had lower min-matched benefactors. So to sum up this part, we've shown that consistent with the prediction of probabilistic inference in a GSM, surround stimulation reduces response variability more than it reduces response strength. And this suppression variability goes beyond the suppression expected just from receptive field stimulation. Now, another interesting and an advantage of this neural sampling hypothesis is that you can very straightforwardly go beyond single neuron phenomena. And so, for example, we can now look at the structure of the joint posterior distribution of two latent features represented by two neurons. And so we have, again, this kind of generic prediction that the covariability between two neurons, their noise correlations, should reflect the inferred statistical dependency between latent features. So for example, if the latent features are correlated in the posterior, then we would expect large noise correlations between the activity of the neurons, and vice versa, no noise correlations if there is no correlation in the posterior distribution. So once again, we can now study the structure of this posterior distribution and use it to generate predictions, very specific predictions that then we can test experimentally. Now, the additional complication here is that we don't just have this small image, large image condition, right? It's not just controlling the stimulus context, but there are additional considerations that we have to make about, for example, about the properties of the two neurons, right? Are we looking at two neurons that are selected for the same orientation and the same spatial locations? Or are they selected for different spatial locations or different orientations and other, dimensions of the stimulus? And all of this will, as I will show you, generate different predictions for effects of context on covariability. And similarly, we have to take some decision about how, about the generative model, right? So for example, we could use these independent GSMs to represent the inference of one neuron and then of the other neuron. Or we could have a shared GSM where there is a single global modulating factor that is shared between all of the filters representing both neurons. So we started exploring a little bit these, the different predictions, and what I'm showing you here is for a set of natural images, how noise correlation, each one of these panels is how noise correlations depend on the size of the image and on the different orientation between the two neurons, and how these predictions differ for shared and independent GSMs. And I don't expect you to go through all of the details of this, but you can already see a couple of things which are also summarized here when we average across all the images. One is that we have this dependency of noise correlation strength on the similarity of tuning of the two neurons, which is a well-known property of noise correlations in cortex. And the other is that the effect of stimulating the surround with a large image of the effect of contextual information is qualitatively different, in fact, opposite when you assume a shared modulator versus independent modulator. In particular, with shared modulator, the prediction is that contextual information will reduce noise correlation between the neurons, and whereas it will increase it for the independent. And so the intuition for this really is that in the shared modulator case, because the modulator is shared and it is unknown, our uncertainty about the modulator is what induces variability, and that variability is shared between neurons. But when we provide more contextual information with large images, we reduce that uncertainty, and therefore we are reducing a source of shared variability. On the contrary, when we have an independent modulator, which is again the main source of variability, that is a strong source of independent variability. And therefore, when we provide contextual information and reduce uncertainty, we now have reduced a source of independent variability, which will let the other sources of shared variability emerge, right? So this is roughly the intuition. That leaves us, though, with a question of, well, what is the better model for any given pair of neurons that we could record from experimentally? And so to start addressing that, what we did was to train a bunch of these GSMs, independent or shared, for pairs of model neurons with different spatial positions here in this matrix and different orientation preferences, and compare which of the two was a better model of the statistics of natural images. So again, we train these models on a database of natural images, and then we compare the log likelihood of the images under the shared model versus the independent model. And I guess consistent with intuition, what we find is that for pairs of neurons that largely overlap in space, the shared modulator is a better statistical model. Whereas once we have neurons model neurons whose receptive fields are completely non-overlapping up in the corners here, then the independent GSM is a better model of the statistics of images. And this is true across different orientation differences for the two neurons in the pair. But of course, for the more difference there is between the two neurons, the more the independent GSM is a better model of image statistics. So why is this interesting? Well, this is interesting because now this provides us with a way of kind of predicting, at least qualitatively, whether a given pair that we record experimentally should follow the prediction of a shared GSM or an independent GSM. So for example, when we're looking at pairs of neurons whose receptive fields are overlapping, we expect that their modulation of noise correlation should follow that predicted by a shared GSM, that we should observe a reduction of noise correlations. And so once again, we went to the lab of Adam Cohn to design experiments to test this prediction. And what, long story short, what we find here is that consistently noise correlations for pairs of neurons with overlapping receptive fields those noise correlations are reduced by stimulation of the surround. On the other hand, for pairs of neurons with non-overlapping receptive fields, of which we have some, thanks to the fact that the array of electrodes is a planar array, and thanks to the fact that neurons that are separate in cortex also have separate receptive fields in visual space. So for these pairs of neurons, we expected that the noise correlations should actually increase when we stimulate the surround, and that's what we find in many of the recording pairs, of the pairs that we recorded from. So at this point, just I want to do a little detour away from normative models. Because at this point, right, we have this qualitative prediction that we can validate with measurements of noise correlation. But one interesting question is how can we more quantitatively tell whether a given pair of neurons actually shares a common normalization signal or whether they are independent? And so for this purpose, and this is the little detour, we developed a class of parametric descriptive models with these two goals in mind. First of all, parameterize and quantify the relation between divisor normalization and variability, covariability. And then also to use these models to be able to estimate single trial normalization strength, which is a quantity that we cannot really measure experimentally. So we introduced this model in a paper that we published a couple of years ago, where it's a very simple extension of the normalization model, except that now both the numerator and the denominator of the model have noise. and their ratio represents the single trial response of a neuron. And we developed this model in the paper and show that with certain approximations, you can derive closed form expressions for the mean and variance of this model neuron and how they depend on the parameters of the variability of the numerator and of the denominator. Therefore, we can have a quantitative relationship between the strength of normalization, the variance of the normalization, and how those things affect the response strength and response variability. We have validated this in a bunch of different experimental data sets, including in macaques and in mice, with size manipulation and with contrast manipulation. I will not go into the details of this. We also validated the single trial normalization inference, which I'm not going to really talk about today. But then we also developed an extension of this to pairs of neurons, and this is now relevant to what I was telling you earlier about sharing normalization signals. Now, in this extension, the noise in the numerator of the two neurons can be correlated, and the noise in the denominator of the two neurons can also be correlated. And these are, in this model, the two sources of noise correlation in the measured responses, right? You have numerators that are correlated and fluctuating, denominators that are correlated and fluctuating, and each of those contribute to noise correlations of the neurons. Again, we have derived analytical solutions for the noise covariance matrix in this model, and we have shown that you can, optimize this model, the parameter of the model, to fit neural data. These are data that were collected in the lab of Hillel Adesnik with calcium imaging in mouse V1. And what I'm showing here, just a couple of example pairs of neurons. The data on the top, where we manipulate the contrast of the stimulus, we see an increase in the mean response of the neurons, and also a change in the noise covariance structure, the ellipses. For this pair, there are positive correlations that increase with contrast. For this pair, there are negative correlations that decrease with contrast. And we see that the model can capture those quantitatively very accurately. But the interesting thing is that we are able to show that for many pairs, pairs that satisfy certain conditions, we can actually identify very accurately the parameters of these, the correlation parameters of the numerator and denominator. And this is here showing a scatter plot of the inferred correlation parameter for the denominators. In black circles are the pairs of neurons for which we can accurately estimate that correlation parameter. And we see that most of the cases have positive correlation between the denominator, suggesting that these pairs of neurons share their normalization signal, that they're part of the same normalization circuit. And by the design of these experiments, these were all pairs were of neurons with similar receptive fields, both spatially and in terms of tuning. So this is something that kind of was alluded to and suggested by a lot of previous literature, that normalization is shared between neurons with similar tuning, but was never really quantified. And with this method, we can now actually put a number on this sharing of normalization signal. And so, going forward, we're planning to use this method to sort of more precisely quantify the predictions of our GSMs for noise correlations. So I'm going to stop here and just go through a brief summary of what I told you. I've proposed to use this normative approach to study how complex visual inputs are mapped onto the statistical structure of neural responses in visual cortex. And in particular, I've proposed to take the view of probabilistic inference as the computational goal of neural populations in V1, and neural sampling as a theory for how those probabilities are represented in activity of neurons. And I've shown to you that within this framework, we can unify two sets of widespread cortical phenomena, including contextual modulation and structured response variability. We can show mathematically that there is a precise role for division normalization in probabilistic inference, therefore linking a very specific computational goal with a phenomenon for which we are starting to really understand the circuit mechanisms, and so potentially offering a link from computation to mechanisms. I've shown to you that there is a diverse range of predictions for how contextual stimuli should modulate the covariability between neurons, some of which we have initially validated with our experiments. I've also shown how the theory predicts that V1 should be sensitive to segmentation cues, which again we validated experimentally, and we've also worked recently on how the framework predicts interesting cortical dynamics for sampling from bimodal distribution. This is something I have not discussed today, but it's other work that's ongoing in the lab. And lastly, I've in the last couple of slides I showed to you how this normative framework can also kind of motivate the development of new statistical tools for data analysis to answer very specific questions posed by this normative framework. So it's been over 45 minutes, so I'll stop here. I'm going to skip the segmentation part, and instead I just want to go to the acknowledgement slide over here to make sure that I give credit. In particular, the first part of the talk was, as I said, work that I did in collaboration with Adelia and Peter and Adam, and the second part on what was done mostly by Dylan Festa, formerly a postdoc in my lab, and then more recently by Amir Farsmadi, postdoc in my lab, and Oren Weiss, also a student in my lab, and in collaboration with Alan Cohn. Thanks to my funding sources, and thanks to you all for your attention.